All right, everyone, welcome. Today, we're going on an incredible microscopic expedition. I mean, we are literally shrinking ourselves down to explore one of the simplest, but also one of the most powerful and abundant forms of life on our planet, the prokaryotic cell. You ready? Let's dive in.
So, to get our heads in the right space, let's kick things off with a bit of a riddle. What could you, a mushroom, and a tiny little bacterium possibly have in common? I mean, on the surface, it sounds like nothing, right? A person, a fungus, a microbe. But believe it or not, you all share the same fundamental toolkit for life.
And here's that toolkit. It's wild when you think about it. Every single cellular organism on this planet, from you and me to the bacteria living in our gut, is built using these four essential components. You've got your DNA, which is the master blueprint. The cell membrane acts as a bouncer, controlling what comes in and out. The cytosol is kind of like the jello filling up the inside, and the ribosomes, those are the little factories building all the proteins. It's literally the universal starter pack for life.
Okay, so here's our game plan for this journey. First, we're going to nail down what a cell even is. Then, we'll take a tour of the outside, break through the cell's defenses, peek inside the control center, and finally, see how these amazing little machines make more of themselves.
All right, Section 1. What makes a cell? Seems like a simple question, but the answer takes us right to the biggest, most fundamental split in the entire tree of life. We're talking about a division that happened billions of years ago and basically set the stage for all of biology as we know it.
And here's that great divide. It really all boils down to one thing: the nucleus. Eukaryotic cells, that's us, plants, fungi, we have a true nucleus. It's like a secure vault, a membrane-bound room just for our DNA. But prokaryotic cells, the ones we're looking at today, their DNA just kind of hangs out in a region called the nucleoid. There's no wall around it, and that one little difference, that's everything.
To really understand what a cell is, it helps to know what it isn't. And that brings us to viruses. These things are acellular. They're not even real cells. Think of them more like biological pirates. They're just a bit of genetic code wrapped in a protein coat, and they can't do anything on their own. They're completely helpless until they can hijack a real cell and force it to make more viruses.
Okay, tour's officially starting, and we're beginning on the outside of the prokaryotic cell. We're going to check out all the cool gadgets it has for moving around, grabbing onto stuff, and defending itself from a pretty hostile world.
So a lot of bacteria rock this thing called a glycocalyx, which is basically a sugar coat. And it comes in two main styles. First, there's the slime layer. It's loose, it's gooey, and it's perfect for helping bacteria stick to surfaces. That film you get on your teeth in the morning? Yep, that's it. But then you've got the other kind, the capsule. This one is neatly organized, tough, and it's a serious piece of hardware. It acts like an invisibility cloak, helping the really nasty bacteria hide from our immune system.
And what about the attachments, the things sticking out? Well, bacteria have this amazing toolkit for getting around. You've got flagella, which are basically little propellers, like an outboard motor pushing the cell along. Then you have these super cool axial filaments on spirochetes that act like a built-in corkscrew, letting them drill right through tissue. For just grabbing on, they have fimbria. Think of them like the bristles on a brush. And then there are pili, which are incredible. They're like little grappling hooks that they can use to connect to another bacterium and, get this, transfer bits of DNA.
Okay, so we've seen the external gadgets. Now it's time to go deeper. We're about to breach the cell envelope. This is the really critical part. It's the cell's armor, its walls and its gates all rolled into one. This is what separates the chaos of the outside world from the order inside.
And right at the heart of that armor is this absolutely incredible molecule you won't find anywhere else in nature. It's called peptidoglycan. Just imagine a super strong, flexible mesh made of sugars and proteins all linked together. It's like molecular chainmail. This stuff is what gives the cell its shape and, crucially, keeps it from literally exploding from internal pressure. It is the key to a bacterium's survival.
Now, how that peptoglycan is arranged lets us split most bacteria into two huge groups. On one side, you have gram-positive bacteria. They have a really thick, exposed wall of peptoglycan. It's like they're wearing their chainmail on the outside. On the other side, you have gram-negative bacteria. Their peptoglycan layer is thin, and it's sandwiched between two cell membranes. This difference might sound small, but it's absolutely fundamental in microbiology. It changes everything, including how we design antibiotics to fight them.
And we're in. We've made it past the walls. Welcome to the cytoplasm. Think of this as the bustling factory floor of the cell. This is where all the action happens, where all the chemistry of life is going down.
So what's on this factory floor? Well, smack in the middle, you've got the nucleoid. That's the main control area holding the big circular chromosome. Then you have these little things floating around called plasmids. They're extra little circles of DNA, kind of like carrying a specialized tool or a secret recipe, often for things like antibiotic resistance. See all those tiny dots everywhere? Those are the ribosomes, the actual protein-building machines on the assembly line. And finally, you've got inclusion bodies, which are just storage closets, places to stash extra nutrients for a rainy day.
Okay, we've seen all the parts, we've seen the factory, so what's the whole point? Well, it's to do one thing: make more factories. Let's take a look at the beautifully simple and ridiculously powerful way that bacteria multiply.
The process is called binary fission, and it is elegance itself. It's just 3 steps. Step 1, the cell makes a perfect copy of its circular DNA. Step 2, the 2 copies get pulled to opposite sides of the cell. And step 3, the cell wall and membrane just pinch inward in the middle until, poof, one cell becomes 2 identical clones. It's simple, it's fast, and it is unbelievably effective.
And if you watch a whole population of bacteria in, say, a petri dish, you'll see their story unfold in 4 distinct acts. First is the lag phase. They're just getting settled, tooling up for growth. Then comes the log phase. This is the explosion, exponential growth, where the numbers just skyrocket. After a while, they hit the stationary phase, where things level off. New cells are being made at the same rate that old cells are dying. And finally, as the food runs out and waste builds up, you get the death phase, where the population declines.
All right, so to pull this all together, here is the final showdown, the big comparison. For prokaryotes, no nucleus, no complex organelles, that unique peptidoglycan cell wall, slightly different ribosomes, and they multiply by simple binary fission, not that complicated mitosis dance we do. If you remember these points, you've absolutely nailed the key differences. So I want to leave you with this one last thing to chew on. We spend all this time calling these cells simple. But think about it. They have no brain, no nerves, nothing we'd call intelligence. And yet, they can build complex communities, they can outsmart predators, and they have literally changed the face of our world. What does that tell us about the nature of life itself? That something so simple can achieve so much. It's just incredible, isn't it?